So my name is Mahmoud Moradi. I'm an assistant professor at the Department of Chemistry and Biochemistry, University of Arkansas. And I will be talking about some methodological and also application work that we've done with blue waters. We have access to blue waters through the Great Lake Consortium for Petascale Computing, GLCPC, and I will talk about some of the work that we've done with our allocation. So my talk will consist of a little introduction about larger scale conformational changes of proteins, especially membrane proteins. That's the main topic of the research in my lab. And the methodology that we use is mostly a combination of pathfinding algorithms and free energy calculation methods, and both of these categories are using, at least the way that we do it, petascale supercomputing, in the sense that we use loosely coupled multiple copy algorithms that use hundreds to thousands of replicas of the same system simulated at the same time to be able to find the most relevant pathways and also thermodynamically characterize them and calculate free energies for them. There has been some theoretical work done in the lab to improve these algorithms at the theoretical level, and I will mention that a little bit, and that is the Riemannian reformulation of some of these algorithms. And finally, I will talk about the application, which is for the proton, basically the title of the talk, which is the Proton-Coupled Oligopeptide transporters, or POTs, a specific class of membrane transporters. So membrane transporters and also other membrane transport proteins, such as channels, at least some of them, rely on larger scale conformational changes. And these conformational changes are very slow. They usually occur on millisecond level. Sometimes it takes tens of milliseconds, hundreds of milliseconds, and in some cases even seconds for a particular transition, a larger scale conformational change to happen. Because of that, regular molecular dynamic simulations are not enough to help us study these conformational changes. And actually, my talk starts with a case study to see what happens if you try to use regular molecular dynamics equilibrium simulations to try to understand the mechanisms of such systems. So this example is exactly the topic of this talk, which is a proton-coupled oligopeptide transporter, or a POT transporter, specifically GkPOT, which is a bacterial POT transporter. So what I show here is a particular 400 nanosecond simulation. And this is very typical. This is actually a very common practice. You pick a crystal structure of your protein of interest. You are interested in learning about its mechanism. The mechanism is what determines how these conformational changes happen and how they are coupled with chemical events such as protonation, such as substrate binding, and so on. So you try to do these simulations in different conditions. For instance, you want to protonate a particular residue, which is the putative proton binding site, or you want to dock a substrate, or if the substrate is already in the crystal structure, that's better. So you do the simulations with and without your substrate. So you do this in different conditions, and then you see what happens, what are the differences in these simulations. So in this case, we did that as well. So we did 2 repeats of 400 nanosecond simulations for 8 different conditions: protonated, unprotonated, different substrates, and so on. So this is doable. You don't need blue waters for that. You can do this actually on other supercomputers as well, or even on your local clusters. But the question is really, what can we learn from this? And I'm going to show you how difficult it is to actually learn anything from this kind of simulation. So what you see here is an animation, a movie of a simulation like this, which is interesting. You can attach that to your manuscript that you're submitting, and it's cool. But let's see if we do statistical analysis of our results, what happens. So what I've done here, I'm showing you a number of different measures that we use to characterize the different global and local conformational changes. For instance, this is a local measure. It's the distance between two different particular amino acids that form salt bridge. So we are interested in knowing how the salt bridge distance changes, whether we have salt bridge breaking or formation. These are local conformational changes that may or may not happen. We also have more global conformational changes, such as the relative orientation of these two particular helices or two different bundles of the protein. They may or may not change depending on the condition. So you can characterize the larger scale motions, and sometimes people use simpler measures rather than looking at the interdomain rotational changes, interdomain orientational changes. They just pick two, say, loops and look at their distances, and they measure the larger scale conformational changes like that. That's also possible. So when you do that, the local conformational changes usually can be characterized, and you will see different behavior in different conditions. For instance, if you do have a protonated versus unprotonated, you see a different behavior. These are local conformational changes. So you can study local conformational changes when you have proteins simulated in different conditions. But what about global conformational changes? So here I'm looking at the interdomain angle between the two bundles of the protein, and also the distance between the two loops that I showed. They should kind of correlate. So if you run these simulations in two different conditions, in this case I have an apoprotein unprotonated and a protonated, particular residue in the protein has been protonated, and there is a particular substrate which has been docked to the protein. So you see two different conditions, and they seem to behave differently. So this is a common practice that people do this kind of simulations, change the condition, and look at the behavior of the system and response of the system. Unfortunately, if you repeat the simulation with the exact same conditions, you may not see the same thing. And this is something that may not be reported in a lot of studies. It is more common to report the differences. When you have different conditions, you see different global behavior, and you have a story to publish. But if you try to reproduce this, this may not actually necessarily happen. And here, we only repeated this twice. So it was just two repeats of the same simulation. And interestingly, it happened to behave very differently in one case and behave very similarly in the other case. That's just a coincidence. And coincidences usually don't actually mean anything in a lot of molecular dynamics simulations. If you want to analyze this in a more statistical manner, then you will actually see that these differences are not necessarily meaningful. So, okay. This is actually not my work. This is just a figure from a paper that I've cited here. This is the exact same thing that was reported in that paper. They did the exact same simulation that I told you about. They ran a 200 nanosecond simulation in a bunch of different conditions, and they observed a different behavior for the global conformation of the protein, and they reported that. So this happens a lot. However, although this is a common practice, but statements made above mechanisms of proteins that work on millisecond level. So the molecular processes that occur on millisecond and second level, you cannot make statements about such processes if you are running equilibrium simulations that are sub-microsecond. So this is what we learn actually from this kind of simulations, that maybe we should be a little more humble about what we learn from molecular dynamics simulations. And if you do this in a more statistical manner, just try to see whether or not different repeats of the same simulation are different or similar as much as the simulations performed in different conditions, you will realize that there is really not much distinction between them. So if you repeat the exact same simulation, you may see a different behavior. And if you run this in a different condition, again, you may or may not see a different behavior. And we shouldn't jump into conclusions that if we see a different behavior, that's because it is the difference in the conditions of the simulations that has resulted in that. Okay, so now that we know we cannot just rely on regular equilibrium simulations, what do we need to do? We really need to do the transition. We need to see the transition. If you want to make a statement about the transition, a larger scale conformational change, you need to simulate that larger scale conformational change. And if your equilibrium simulations are not capable of doing that, you need to find a way of still simulating that. And then maybe you can make statements about this transition. If you don't see the transition, you cannot make a statement about it. That's the conclusion that I can make. So we use non-equilibrium pooling simulations as our first step. So we cannot see the transition happening spontaneously, so we force that to happen. However, what we do that is we try many different protocols. We try different ideas of how to pull the protein to reach a different state. And after that, our pathfinding algorithms and our free energy calculations that are more automatic start. So the strategy basically relies on first kind of an empirical search for different protocols to induce a transition and verifiably get a model for the transition and then optimize that model for the transition using pathfinding algorithms and then characterize that pathway that you find using free energy calculations. And for the pathfinding algorithms and free energy calculations especially, we use algorithms such as string method with swarms of trajectories and bias exchange umbrella sampling. And both of these algorithms are actually loosely coupled multiple copy algorithms that use hundreds or even thousands of replicas of the system. So I'm going to skip this part. So one improvement that we have been working on is to somehow combine the free energy calculations and pathfinding algorithms in an iterative manner. So once you do your pathfinding algorithm, you converge to a pathway, you can do free energy calculations, and then if you analyze your free energy calculations and try to statistically examine whether or not you have converged to the same pathway that you started with, you may actually observe deviations, which then may require more pathfinding, more iterations of pathfinding algorithm, and then you may end up with a different pathway that you initially thought was correct. So there are false convergences that may happen in any of these algorithms, and iteration would always help with figuring out the systematic issues with the convergence. And this is some data that I'm showing here on NAMD benchmark. This is, I think, an older version of NAMD 2.11 that I used back then. And this is mainly to show that this kind of algorithms, the loosely coupled multiple copy algorithms, are having a very good weak scaling behavior the strong scaling may not be as great as weak scaling. So that means that the more the number of replicas that you are using, the more efficient you are actually using the supercomputer, and this is especially appropriate for supercomputers like blue water. So the algorithms scale very well with the number of copies, number of replicas of the system. And this is, by the way, not an embarrassingly parallel algorithm. You have communication between the replicas. It happens every 1,000 steps or so. Okay, so this was an example that we actually did with blue waters a few years ago when I was still at the University of Illinois working with Ahmad Tajkhurshid, that we used this algorithm and that I had just developed back then and optimized it for blue waters with the scripting capability of NAMD, and we studied the full transport cycle of a membrane transporter, which is kind of similar to the POT transporters, but it's a phosphate transporter. So the results were interesting. We could actually characterize the pathway, we could characterize the free energy, and so on. However, what was missing, and I realized later, was the fact that there was some flaw in the whole formalism of pathfinding algorithms and free energy calculations, in the sense that the Euclidean assumption of the free energy landscapes and pathways when you are using with collective variables is actually not a good assumption. So it turns out that if you use a particular set of collective variables for your free energy calculations or for your pathfinding algorithm, you may end up with results that are not invariant under coordinate transformation. So that means that if you find the pathway, that pathway actually would change if you go from one set of collective variables to a different set of collective variables. If you take the exact same collective variables and non-linearly transform them to a different set, you will end up with a different pathway. So the pathway is not invariant, and that actually calls into question its importance. So what I did, I reformulated the entire formalism for free energy calculations and pathfinding algorithms, and it was actually quite easy to implement that with the bias exchange umbrella sampling and a string method with swarms of trajectories methodology, and with the scripting capability of NAMD, so you do not actually need to change anything in the code. All you need to do is the script that is used to manage the communication of these different replicas can be easily altered to go from a Euclidean formalism to a Riemannian formalism. And this was something that we did, and it worked just fine. And we used this, and we are using this now for the POT transporters, particularly for GkPOT, and we have been able to first optimize our initial biasing protocol, find the biasing protocol to go from the known inward-facing state of the protein to an unknown outward-facing of the protein, and we were able to find a good protocol that results into an actual outward-facing state. And we know that it's a relatively good starting point because it actually shows an outward-facing behavior. If you equilibrate this model that we have generated, it shows a water density profile which is actually consistent with an outward-facing state. If you just use a simple modeling approach to come up with an outward-facing state, it may look like an outward-facing state, but when you equilibrate such a model, it may not actually happen to be stable. It is very easy for it to become occluded, for instance, because these proteins kind of like the occluded state. It's very natural for them to get to the occluded state. But if you end up with a protein which is stably open, that is actually a good sign that you have a good starting point. And after that, we can do path optimizations with string method with swarms of trajectories. And if you start with an initial good path, your string method with swarms of trajectories is going to converge faster. And if you start with a bad path, the convergence that you get from your pathfinding algorithm is probably a false convergence. You have to start with a good path if you want to actually learn something about the system. These algorithms are just optimization algorithms. And what I'm showing here is just the two trajectories, one coming out of our best non-equilibrium protocol, and the other one is the optimized one, which basically, if you see that the closing in the intracellular site happens, the opening on the extracellular site happens, and there are some differences. I'm just highlighting one in the two. So the string method with swarms of trajectories is good for optimization of a pathway that is already a good pathway. So this concludes my talk. These are my lab members who have done most of these simulations, and I would also like to thank the Blue Waters and the Great Lake Consortium for petascale supercomputing, and for also the funding agencies and so on. Thank you very much.